My research involves bioenergy, and I'm going to dash out into an area that I typically don't get involved in, which is social justice, and see how I think bioenergy can actually impact social justice and improve society on the whole. So to get started, first of all, we need to understand what both bioenergy is and also how social justice, at least as I'm interpreting it, comes into play. So bioenergy is renewable energy derived from biological sources. So we're going to hear later on today about biofuels from algae, from Dr. Suksadu, microbial fuel cells, direct electricity production, or even more recently you may have heard about biofuels, bioethanol from corn, as we've had the discussion about should we be using it for fuel or should we be using it for food, especially with the drought that's been going on recently. So that's another area of bioenergy that we can explore. And then social justice, the way I'm looking at it is social justice is justice exercised within society amongst the classes. And in particular, I'm thinking about developing countries and how we as a privileged society can support them and help them to improve their lives. We already have a sense that bioenergy can improve society. With the advent of the Industrial Revolution about 200 years ago, we started to improve society, but that was done using fossil fuels. And the fossil fuels brought in carbon that's been long buried. And as a result of that, there's been pollution in terms of soot and also carbon dioxide, which has caused elevation in carbon dioxide levels and resulting in global warming. Bioenergy, on the other hand, uses carbon that's actively involved in the carbon cycle, and so as a result, there shouldn't be this increase in carbon dioxide levels, and hopefully a leveling off of those. And so we should see cleaner water, cleaner air as a result of these improvements. So we've got a lower environmental impact from bioenergy across all of society, but we've got some other advantages associated with it as well. One option is localized production. So rather than bringing the coal from maybe West Virginia a few hundred miles to a power plant for combustion, or petroleum a few thousand miles away from the Middle East, we can produce our energy locally and use that to support the community. One of the reasons this is possible is because of the diversity of energy that's available from biosources. So what may work here in Ohio may be different from what works well in Indiana, which may be different from what works well in Illinois. And so each local region can have their own specialized biological sources of energy. So, that's great for the world on the whole and for developed countries, but what about developing countries? What kind of benefits can they gain from bioenergy? Well, looking at the literature, it seems like there's three things that developing communities need. They need clean water, they need food, and then they need energy. Biotechnology certainly can address all three of those. I'm going to focus on the energy aspect with this. And I see a few different energy types that can support development of these communities. One would be electricity. So with electricity, we get improved communication, maybe operation of radios for warnings. Also improved quality of life through lighting, allowing staying up and continuing to work later into the evenings if desired. We can also make biofuels, which can improve transportation, resulting in increased trade. Supplement heating and cooking oils to improve those, or with use with the generator, be used to produce electricity. So a number of different ways that I see bioenergy being able to support these developing communities. Well, what types of energies are available out there from biological sources? Biofuels is a big one. Again, we've got, that's better. We've got biofuels that can be converted into diesel using algae, oils from algae or other sources. Bioethanol that can arise from corn, or a next generation arising from cellulose. So I'll talk a little bit about that shortly. biohydrogen. So you may have heard about the hydrogen economy. This was big about 5 or 10 years ago in terms of moving to this clean energy. When we react hydrogen, it makes just water. There's no carbon dioxide given off. Well, hydrogen can be made through electrolysis of water, generating hydrogen and oxygen, or it can be produced by microorganisms as well. So we can utilize the natural metabolism of the organisms to make hydrogen. One of the areas I'll spend a bit more time talking about in a few minutes is microbial fuel cells. This results in direct electricity production from the biocatalyst, similar to other fuel cells, so the hydrogen or methanol fuel cells. And then finally, fuel oils, which are related to the biofuels themselves, which would typically be used as additives for cooking or heating oils, and are derived from biological sources, similar to biodiesel. So if I could take a few minutes to tell you a little bit about the research in my lab and how I see some of this fitting in here. The research in my lab looks at some bioenergy applications and has two major thrusts associated with upstream processing of biofuels and also microbial fuel cells. So the first aspect I'll talk about just for a minute is upstream processing, and this uses polysaccharides such as starch or cellulose, and we're focusing on cellulose right now, to provide the monosaccharides that can then be fermented into ethanol. So starch and cellulose are both made up of the same monosaccharides, glucose. And if we break those down into glucose, that can then be converted into ethanol through yeast. To get there, it requires a lot of energy, typically high temperatures and chemicals to break down cellulose. We're looking at biological mechanisms to achieve the same process. So how do we use enzymes to break down cellulose into oligosaccharides and then into smaller oligosaccharides and finally into the mono and disaccharides? So we can use enzymes such as cellulases and beta-glucanases and finally beta-glucosidases to generate this. We're using organisms that are called hypothermophiles as the source of these enzymes because they grow at high temperatures. So these are organisms that grow at 80 to 100 degrees Celsius, so boiling water. This gives us a few advantages because at higher temperatures, sugars are more soluble, so we can get a higher concentration and hopefully a faster reaction rate associated with that. We also get reduced risk of contamination. So as we're breaking down these sugars and generating glucose, just about any organism would love to get its little pili on those. So if we can prevent those organisms from starting to grow in it, it'll give us more energy to convert into that. So at the elevated temperatures, there's fewer organisms for contamination. The other area that we've been looking at is with microbial fuel cells, and this has been a collaborative project with Dr. John Rowe in biology and Dr. Andrew Sarangan in electro-optics. And this uses direct electricity production by microorganisms. So it's similar to hydrogen fuel cells or methanol fuel cells that you may be familiar with. In the case of a hydrogen fuel cell, it takes hydrogen and it splits it into hydrogen protons and electrons, and the electrons, of course, are the electricity. Now, in the case of methanol and hydrogen fuel cells, those require the specific type of fuel source. Even using a similar fuel source like ethanol in a microbial fuel cell isn't terribly efficient. Microbial fuel cells, on the other hand, use microorganisms as a catalyst to process the energy sources. So process glucose or some other polysaccharides or organic acids. And because of the evolution of the organisms, they're able to utilize a wide variety of energy sources, so we're not limited to just one single type. So that's one of the advantages associated with this. And we've been looking at microbial fuel cells at both bench and small scale, and using a number of different organisms for these studies. So you can see a picture of it here on the left, and I won't go into too much detail, but we've got a side called the anode, which is where we process our energy source. So in this case, we're showing glucose as our energy source, and this is where the microorganisms typically are utilized. Right-hand side is the cathode, where we take the hydrogen and convert it into water, and we'd still generate protons in this case. One of the things we've been doing in the lab, and this has been the collaborative project with Dr. Serenga and Dr. Rowe, is trying to miniaturize microbial fuel cells. This isn't something that would necessarily be implemented in a developing country, but it would be the size of maybe a 9-volt battery or something like that, where we take it and have our anode and our cathode fluids, so these are the two sides of the chamber, flow next to each other. And instead of being separated by a physical barrier, they're separated by fluid flow properties, maintaining a laminar flow regime in between those. And so we designed a two-channel system, and you can see some of the results. We've got a dyed region on the left here and a buffer region on the right. And as it flows down, we still have separation between the two anode and catholytes. This was very promising and we were very excited, so we decided to see if we could expand this and increase our energy density and our density of our system by going to a 6-channel system. And so this is based upon a similar approach, alternating our anode, cathode there in red or in yellow and silver, and still having the same flow maintained by separation of the flow properties. You can see here on the right that as it flows down, we do have this continued separation, and so we've got buffer dye, buffer dye, buffer dye. And at the feed region, looking at this, we've got our dyed region that we're looking at here, so we've got good separation. There's no breaking between the barriers, good integral design. We've got some support here so the channels don't collapse, but we're looking at a solid design that's able to generate these laminar flow systems. So we're very excited about this and being able to produce a laminar flow microbial fuel cell that has multiple flow channels. And so this has really been a collaborative project, drawing upon each of our different disciplines, and should lead to what it should be the first multi-channel laminar flow fuel cell.